As you can tell, I'm not Harleen Hayne. She sends her apologies. She's suddenly been taken somewhat unwell. And we also have apologies from Paul Brunton, the Pro-Vice-Chancellor of Health Sciences. So we're a slightly diminished group, but we're still here for this celebration for Richard Macknight. So for those of you who don't know me, I'm Pat Cragg, and I'm the Deputy Vice-Chancellor Academic at the University of Otago. And it's going to give me great pleasure to open this inaugural professorial lecture for Professor Richard Macknight. On behalf of the university, I'd like to begin by extending a particularly warm welcome to the members of Richard's family who are here with us this evening. His wife, Catherine Day, Professor Catherine Day, and their son, Simon, welcome. Richard's parents, Emeritus Professor Tony Macknight, and his wife, Dr. Jocelyn Macknight. Prestigious family line. And we also have at least 3 of the siblings. I can see some of them, yes, and various partners and members of their family. So it's really wonderful that Macknights and their extended family could all be with us today. The presence of family is a clear signal of the importance of these types of occasions. And of course, I'd also like to acknowledge and welcome staff and students from across the university and members of the wider Dunedin community. And for many of these inaugural professorial lectures, the Chancellor, Dr. Royden Somerville, right at the back, thank you again for attending these sessions. These IPLs are always a time of celebration and a pride for the University of Otago. So I'm delighted to see so many people here to mark this important milestone in Richard's career. To be promoted to professor, as Harleen Hayne will always say, is no mean feat. You have to be outstanding, you have to show leadership, and you have to do it across teaching, research, and service. As you will learn this evening, Professor Richard Macknight is a passionate teacher and a popular postgraduate supervisor. He is also a successful and well-funded researcher, and his research career has been marked by success in both the basic and applied projects. And this is in the field of plant biology, plant biochemistry, and plant genetics. Throughout his career, he has provided exceptional service to his discipline, his academic department, and to New Zealand more broadly. His expertise in plant genetics is highly sought after by multiple professional bodies, too many to mention, but I will mention the Royal Society of New Zealand and the Auckland City Council. Last, but certainly not least, Richard is an extremely collegial member of our academic community. So Richard, on behalf of the University of Otago, it gives me great pleasure to congratulate you on your very well-earned promotion to Professor. I'm very much looking forward to your lecture, and I'm now going to call on Emeritus Professor Frank Griffin to tell us a bit more than I have about Richard's academic journey to Professor.
Kia ora. Acting Vice-Chancellor, distinguished academics, fellow academics, friends and family. It is an honour for me to be here this evening to give you a little biosketch of Richard's activity for those of you who are not familiar with the scope of his work. So we're going to hear tonight how plants know when to flower, which sounds very romantic and quite exciting. But what I want to do is give you the chronology of his academic development. And it's lovely to see a professor here in the University of Otago gown. I remember when I came here, there were very few Otago gowns ever in evidence. And it's lovely now to see you've replaced most of the overseas funnies with local graduates, and that's wonderful. So Richard grew up in Dunedin, and he studied botany and biochemistry at the University of Otago. He graduated BSc Honours in Biochemistry in 1990, and then did his PhD, which he completed in 1993, both working with Associate Professor Kevin Farnan. Obviously, he had a very good training in that area. He then left Dunedin and went on the typical overseas postdoc, and he went to the John Innes Centre in Norwich, where he acted as postdoc between 1994 and 98, working with Caroline Dean, who subsequently became Dame Caroline Dean, FRS, one of Britain's most prestigious academics. And that obviously is a mark of his progress, because having a successful postdoc in a high-quality lab is wonderful training for us all when we leave our alma mater. So Richard returned then from Britain in 1998 on a repatriation fellowship by the New Zealand Lottery Science Repatriation Scheme. And he started his own research as part of that fellowship on flowering time in plants, and went on to be awarded a Marsden grant as a principal investigator in 2000. In 2001, Richard was appointed a lecturer in biochemistry, and subsequently he progressed through the academic ranks in the department to become senior lecturer in 2004 and associate professor in 2014, leading to his chair today. In parallel to his academic developments at Otago, he was appointed in 2012 as a senior scientist at Plant and Food Limited on a 0.2 FTE appointment. And this involved him working Fridays for Plant and Food. But it also allowed him to strengthen valuable collaborations between Otago University and Plant and Food. And there was a reciprocal arrangement established by Plant and Food, where one of their scientists had a 0.2 appointment at biochemistry. Hence, we had a continuous link between the two institutions, which was very productive. His funding since 2000 has raised more than $5 million in funds from all the prestigious research funding agencies in the country. Anybody who can get 5 Marsdens over a career is doing an awful lot better than I did. But during the course, he got major funds from Forst on the control of flowering in onions, from Marsden on flowering in legumes, from Agmart on ryegrass hybridization, an MB grant on non-heading in ryegrass, or non-flowering, and numerous grants from local agencies, the OSMS, ORG, and the like. Throughout his career, Richard has enjoyed productive collaborations with Plant and Food, Ag Research, and a number of the major seed companies in New Zealand. His research has involved a mix of fundamental science and the development of applied innovative technologies, some of which have been patented. And since 2015, I'm very glad to say that he's been an executive member of the Agriculture Initiative at Otago, through Ag at Otago, and this year through the development of the new degree, Ag Innovation. In 2013, he won Best Paper Award in the Otago Medical School. He got the Roger Slack Biology Award, and he won the Translational Award in 2016 and an Otago Proof of Concept Award in 2017. In 2018, Richard was elected president of the New Zealand Society of Plant Biologists. Richard has supervised 16 PhD students, 11 MSc students, and 13 honours students. Our delight in Richard gaining his chair is reinforced by exciting prospects as to where plant molecular biology can take us in the future. It's a very, very exciting theme where many of the big questions in biology resolve around plant biology. Increasing plant diversity is one of the most pressing ecologic issues facing our planet. Its success will rely heavily on an improved understanding of the functional biology of our plants and their ability to contribute to sequestration of greenhouse gases and the like. The recent discovery of the plant holobiont is, to me, amazingly exciting, where we now see plants, just like humans, having more microorganisms than host organisms associated with them. And this obviously gives incredible genetic scope to an organism, and hopefully it will inform the research Richard continues to do. So we see very exciting prospects in the future. We're delighted you will be an ambassador for plant biology research. We thank you for your contribution to the university so far, and we look forward to hearing how plants know when to flower.
Thank you very much for the introductions. Thank you, Frank. It's a great pleasure to be here, and lovely to see my family here, so thank you all. And a particular thank to Catherine for all the support over the years that she's given me, and to our son Simon. I know from personal experience it's not easy to be a son of university parents, so it's been an interesting time for him. I remember when Simon was at primary school, he and all the people in his class had to write down what they wanted to be when they would grow up, and it went on a poster outside his classroom. And of course, kids wrote down that they wanted to be a professional netballer, or hockey player or something. Some of the kids wanted to be YouTubers, which I actually didn't know what YouTubers even were. But one boy wrote down Vice Chancellor. So there's a position coming up next year, so who knows what's going to happen. Okay, so in the talk today, I want to talk about how a basic understanding of plant biology and how genes work can lead to developing enhanced crop plants. And my research is involved in looking at a number of examples, flowering, and a number of examples of how understanding the basic biology, I think, is really important. So I'll go through those. So I was born in 1968, and that year there's a book written, The Population Bomb, and on the cover it says, while you're reading these words, 5 people, mostly children, have died of starvation, and 40 more babies have been born. 40 more babies are born. So it was painting a pretty grim picture of the future of the planet, and as came to pass, worried about this big population increase that was going to be seen, and has been seen, and how we're possibly going to feed this growing population. So luckily, there was plant biologists and breeders, and through increasing the production of, and here we've got grain production in this axis, grain production was able to keep up with this amazing increase in population. And the surprising thing was that increase in grain production was done on the same area of land that it was in 1968. So it's all driven through increased productivity, through better breeds of grain, plant breeders, and intensification of agriculture. And we've still got this challenge today. Population is increasing through increased number of adults, and adults living longer. So there's this big issue of feeding the world. And we have to be able to do that on the same amount of agricultural land and in a more sustainable way. So farmers are getting criticised for the pollution and everything, so we need to be more sustainable. And it's a big challenge. So to meet that challenge, I'm passionate about the fact that we need innovation. We need to use every tool that we've got to meet that challenge, to feed the world, especially in front of climate change, which is making growing crops more difficult, and there's more crop losses happening. So I'm very proud to be involved in Ag at Otago, and the fact that Otago is recognising the importance of agriculture and the role that it can play in providing innovative solutions to some of these problems. And this year, for the first time, we have a degree in agricultural innovation. So a really exciting initiative, and I hope we'll train a future generation to try and help solve these problems that are enormous. Okay, so I'll start by just talking about my my research career, and I started working for my PhD with Kevin Farndon. So Kevin's a tremendous supervisor, so personally thank him in front of everyone for the tremendous job he's done. And I'm not the first professor he's trained by any stretch. There's numerous professors that he's been training. My other supervisor was Paul Reynolds, and Paul Reynolds worked at DSIR, which is now Plant and Food Research, so it was great to have an opportunity to work both in an academic institute and in a CRI, and I think that shaped the way my future career went. That collaboration between CRIs and universities is really important, especially given there's not too many academic plant biologists in the country. Paul Reynolds has gone on to have a stellar career after supervising me. So he is Chair of the Board of Ag Research at the moment. He was Chief Executive of the Ministry of Environment and had numerous important roles in science leadership. So he was, I think I've said, might have not, a student of Kevin's in the department, so someone that we're very proud of in our department and the role that he's played. So my PhD was asking kind of a very simple question. Legumes are able to fix nitrogen, and they do so by making these root nodules, and that's made up of plant tissue. So somehow the plant is able to express key genes that are able to make these root nodules. So how are genes controlled and switched on just in the root nodules to make this important structure was the question of my PhD. So here I've got a picture of a gene. A gene has a seat region that encodes a protein, a transcript will be made, messenger RNA, and that will encode a protein, and that will be important in this case for making root nodules. And a gene also has regulatory sequences. And what I was able to show during my PhD was there are certain sequences within a region that is important for expressing that gene just in the root nodules, where a transcription factor is able to bind. And through making transgenic plants, I was able to then introduce a mutated version of that where this sequence wasn't there and show that that was the key sequence for turning on the gene in root nodules. So I've been making transgenic plants for 30 years. It's a fundamental tool for what we do and how we understand biology. It's not a new thing. As Frank said, after I finished my PhD, I was lucky enough to go over and work in the John Innes Centre. It's one of the eminent plant research institutes in the world, and there was just an amazing number of great scientists working there at the time. And the picture here, all this area is glass houses, so huge facilities for growing and analysing plants. Right next door here, it has a cricket field, so I could just walk out the door of the lab and go and play my evening cricket and things, so it was a tremendous place to work. So my supervisor, Frank said, was Caroline Dean. She'd joined the John Innes about 6 years before I came along, and her husband is also a well-known plant scientist, so they turned up at the John Innes Centre wanting to get jobs. And had an interview, both had interviews, and they said to her husband, Jonathan, I'll give you a group leader's job, and then said to Caroline Dean, being a woman, oh, so you want to be a postdoc somewhere, do you? And she said most firmly, no way, I want to run my own group, and quite taken aback, the director there gave her her own group to run. And she's another one, again, after supervising me, has had a stellar career. The other exciting thing at the time was that the first genetically modified plants were just coming on stream, and so we could really see the potential of our research and how it could be translated into new and improved crops. So these flavour saver tomatoes were on sale at the supermarket, and FDA concluded about them that they're as safe as tomatoes bred by conventional means, and so they're allowed. And that's true today as it was then. After 26 years or something of research into genetic modified plants, that statement that they're as safe as crops bred by conventional means is true. And so at a time where we're all meant to be trusting the scientists on climate change and the FDA, whether the coronavirus vaccine is going to be safe, we should be listening to the scientists that genetic modification is safe, and it's a tool that we should certainly be taking advantage of given the need that we have coming up. So what Caroline Dean's lab was working on was how plants know when to flower. So when a plant flowers determines when it produces its fruit or grain, so it's a key agricultural trait and something I thought was certainly worthy of understanding more about. At the time, not a lot was known about the mechanisms of flowering. It was known that plants needed, or many of them, prolonged winter cold and then spring long days, and that will trigger flowering. But exactly how that occurs was not known. It was known that there was a hormone made in the leaves that moved up to the apex, and that was discovered in postulated in 1936, and many PhD students tried to isolate that hormone without any luck at all. So there was a lot to learn. So the way to go about tackling such a problem is through genetics, and I've just there like to acknowledge Russell Poulter. I did second-year genetics with him and got passionate about the power of genetics and its role. I also see Paula Jamison in the audience too. I did botany and she was one of my lecturers and inspired me again to do plant biology, and so I thank her. And she's since become a colleague and we've even published papers together, so it's lovely how things go round. So what it was decided was that it was better for researchers, or one way researchers could tackle a problem is all focus on a single plant rather than looking at their individual plants, focus the effort and get the genetic tools and resources available that everyone worldwide could use. So they chose Arabidopsis, a model plant that can easily be grown in the lab, and it's been ideal for genetic studies. And so the idea was to create mutants, and then if you're interested in flowering time and you've got a mutant that's defective in flowering, then that gene that's been mutated must have some role in flowering. And so by discovering the gene, you should get an insight about how that gene works and the mechanism in general. So I'll talk about 4 different genes here that have provided an insight about how flowering occurs. There's probably about 100 different genes that some ways affect flowering, but here are 4 key genes which we'll go through. So one of those genes, and here's a model that was, a very simplified model that was established. One of those genes is the FT gene, and that makes a transcript which encodes a protein, FT protein, and it turned out, after probably 10 years of working on the FT gene, that actually that was the floral signal molecule, florigen. So the FT protein itself is made in the leaves, moves through the phloem to the apex and triggers flowering. So we can think of now, as a molecular geneticist, I can think of flowering as really when we turn on this FT gene. And so we can go back to thinking about regulatory regions and promoters. And so how FT gene is turned on is through competition between 2 transcription factors, 2 proteins that can bind to the DNA and switch a gene on or off. And so one of these, FLC, is on at high levels in the autumn, and so the plant can't flower in the autumn even though the day lengths are right, because it binds to this DNA sequence and stops the FT gene being on. In the winter, this FLC gene is silenced, it's not active, and so a CO gene can be turned on, protein made in response to the day length, and it can trigger FT and trigger flowering. The gene that I was working on was FCA, that turns out to be a regulator of FLC. So again, like many other people around the world and at the John Innes Centre, we were trying to clone and work out what genes were mutated, and particularly mutants. I was working on the FCA mutant and set about by map-based cloning the gene. At that time, it was a slow process, and the tools for cloning genes were just being established. The Arabidopsis genome was just being sequenced in the lab next door, and so the FCA region was one of the first regions of Arabidopsis was sequenced, so we could discover what genes were there and eventually find out that the FCA gene encoded a protein that has RNA binding domains and a protein interaction domain. So here I've got the FCA gene schematic of it. It makes a messenger RNA translated to make this protein. So what RNA does it bind? What protein might it interact with? So a clue to that came, again, when I was making genetically modified plants. I tried to make this FCA gene express at very high levels. And to my surprise, it didn't kind of work. And rather than getting the full-length transcript being made, I got this truncated transcript. And what we worked out was that the FCA gene is actually able to bind to the messenger RNA as it's being made, make it shorter, and therefore stop too much of this FCA protein being made. So an elegant feedback regulation. So the more and more we study genes, the more and more we find that the regulation, how these genes are switched on and off, is tightly controlled. And so this is a nice example of that. The FCA gene also regulates that repressor of flowering, FLC, as well. And when I made, again, some transgenic plants, just expressing this transcript, I got early flowering, and I got early flowering because it was able to turn off this FLC gene. And working out how FCA turns off that FLC gene has taken 20 years. There's a paper now Caroline Dean has published in PNAS just a few months ago that is starting to fill in that last gap about how it works. So it's a long process, research. So what happens to this FLC gene is in the autumn, it's turned on at high levels, and that blocks the FT gene being turned on. And in the winter, there is a transcription factor that binds to the FLC gene, but in a different region, and actually makes transcript in the opposite direction. And the FCA gene is able to bind to that transcript and recruit a whole lot of proteins to the DNA, which package that DNA in a different way, change the histone conformation. And that way it's packaged turns the gene off. And it turns it off in a way that stays off. And that really solves one of the mysteries about how plants remember that they've been in the cold even when you shift them into the warm under non-inductive conditions. They'll just sit there, and then the minute you put them into longer days, they'll flower. So they've somehow remembered that they've been in the cold a long time ago. And it's through understanding the way that this DNA is packaged that was important. And there have been about 100 papers relating to FLC, and Caroline Dean was knighted because of her work and understanding this regulation. And she was knighted really because having a fundamental understanding of how genes are regulated is not only important for plants, it's important across all areas of plant biology. And actually, the genetic tools that we could use and can use in Arabidopsis meant that she was leading the field in understanding regulation. So I contributed a bit to some of this understanding, and I'd like to acknowledge Becky Laurie. She did a PhD with Caroline Dean, and then had the courage and craziness to then decide to come over to New Zealand to be my first postdoc. So I'd like to thank her publicly again for all the work that she did, and especially starting off in the lab and helping me out and get going. Her work was a tremendous benefit. The other person that didn't work on FCA, but I'd really like to acknowledge, is Rob Day. So Rob Day was one of my first PhD students, worked on a different topic, but he was passionate about technology and innovation and the way we did our experiments, and really led my research forward, and subsequently has got into high throughput sequencing, and it's really a benefit across the university, making this kind of technology accessible. And her and Becky, in fact, were leaders early on in NZGL and our ability to sequencing. So through those two people, making a big impact of what research has been done at Otago. Okay, so how do plants flower in the spring? So we all know that days get longer. So we're up to the 16th of November, and we have 15 hours, or nearly 15 hours of daylight, compared with the shortest day when we only had 8 hours, 40 minutes. So day length gets older. So how do plants sense that increase in day length to trigger flowering in the spring? So insight of that came from actually another New Zealander, Joe Puttrell, who cloned and discovered the CO gene, actually now a professor at Auckland University. And it was discovered, once that gene had been cloned, that it oscillated. And so that at midday, it's very low levels, and at midnight, it's very high levels. And it oscillates, the same way throughout the seasons. It doesn't change. It encodes a protein, CO protein, that happens to be rapidly degraded in the dark. So in the winter, when there's high levels of this messenger RNA, it makes a CO protein, but the CO protein is rapidly degraded, so it can't. It's a transcription factor that activates FT. It's degraded, so it can't activate FT. But when days get long enough, then more of that transcript's made during the light, and it's stabilized and therefore can activate FT. So it's the coincidence of the way that the messenger RNA has been made and the stability of the CO protein in the light that allows plants to flower in the spring. And that mechanism is really precise. You'll all know that plants flower, the same week every year, more or less. So it's a precise mechanism. So a lot was being learned about how flowering occurred in Arabidopsis, and I wanted to know if the same pathways and the same process happened in other plants, and probably because of my PhD, set out studying legumes, an important family of plants, and have collaborated with Jim Weller, University of Tasmania, for many years. He's a fantastic scientist on trying to look and see how flowering is regulated in legumes. I thought it was going to be quick. And what we learned from Arabidopsis would just translate neatly over to legumes, but that wasn't the case. So the key genes that I've talked about, FT is in legumes, and actually there's 2 copies of that, but CO, FLC, and FCA don't play a role in the regulation of flowering. So there's fundamental differences that needed to be discovered. Interesting to discover. So again, some experiments from Becky Laurie. One of these FT genes was turned on in response to prolonged cold days, so the expression level of the gene increases, switched on upon cold, and we were able to get a mutant in this gene, and that plant was no longer able to sense the cold and flower in response to the cold. So it showed us that the cold is able to directly turn on the FT gene, which then triggered flowering. And through the work of Donna Bond, we're able to start getting some insights about how this occurs. So what it turns out is the FT gene is turned off in the autumn because it has these histone modifications, the way it's wrapped up in the DNA. And what happens in the winter is, again, a non-coding RNA is made, and that somehow removes these histone marks and changes the way it's packaged so that in the spring it can be turned on. And so it's really interesting that a similar mechanism, but opposite mechanism, happens compared with Arabidopsis. One plant has a repressor of flowering, which is turned off, and legumes have an activator of flowering, which is turned on, but they still involve this way it's packaged in the DNA and the way it's switched on. So evolution has used common mechanisms to solve that problem. Legumes flower in response to day length as well, and when we put the plants into long days, we get this other FT gene being rapidly turned on. And so it's another trigger for flowering. So it's a combination of these 2 genes that both encode a florogen that determine when legumes flower and the balance of those. And so a postdoc in my lab has been working on this and other things. To my delight, we've now got gene editing working in Metacargo. Shireen also helping make these transgenic Metacargo plants, not an easy thing. And so we've been able to show, I think, results are just coming, that when you knock out this gene, you're later flowering and showing its key role in flowering. But having the ability to mutate genes at will through these new technologies of gene editing is really going to push what we can do forward and revolutionize our ability to do this kind of research. So it's very exciting. Sol also got a fast start Marsden, so it's a very talented scientist, and she's particularly interested in how the clock works and how that changes in the clock are important for adapting to new environments. So we've got some interesting things going on in the lab. I was also interested in the domestication of crops and legumes, and so we've been eating legumes for a long time. Peas, wild peas, chickpeas were in the diet 23,000 years ago, and it was one of the very first crops, along with cereals, to be domesticated in the Fertile Crescent. And the domestication of crops requires a number of steps, but one of them is changing the way the plant responds to the environment so that you can plant the plants in early spring and then harvest them, late in summer, early summer, whatever. And so it requires identifying changes. So through the work of Jim Weller that we contributed to, we found that one of these differences between a wild variety of legume and a domesticated variety of legume was the function of this gene ELF3, which works as part of the circadian clock. And so mutations in this gene lead to a plant that will flower even if it's not in long days. So this early flowering demonstrated in Arabidopsis. So what's happening is that a mutation in the clock alters the pattern of expression of one of these key genes so that it's expressed earlier in the day. And if it's expressed earlier in the day, it's made during the light, even in winter, and will make this stable CO protein, or whatever that gene protein is, to activate flowering and turn on flowering. And in fact, we're yet to discover exactly what that gene is, but we know how it behaves. So you can think of, and I think of, the advent of civilization and moving slowly over time from establishing agriculture on the Fertile Crescent through Europe is a process of waiting for flowering time mutants. So these key mutations that enabled agriculture in new areas and plants to become adapted to new areas slowly happened and slowly were discovered as part of domestication. And that's part of the reason why the move took sort of 6,000 years for civilization to move. And the mutation in the ELF3 gene was key to moving further north in northern Europe. And so getting up into that area was waiting for an ELF3 mutant. And an ELF3 mutant was found allowing peas to be cultivated there and lentils to be cultivated in northern Europe. In chickpeas, there happens to be two ELF3 genes. So the random chance of knocking out both those are too small. So that never occurred in nature. This gene was never mutated. Both copies never mutated to get early flowering. So other gene, in fact, an FT gene was mutated. Which illustrates that now our knowledge of how we can change flowering time and what genes could be disrupted can provide a way of breeding a new chickpea. Unfortunately, with climate change, the seasons are getting shorter, it's getting hotter earlier, and so we need varieties that mature more quickly. And so if we can use gene editing to knock out both copies of the ELF3 gene, we will get an earlier maturing chickpea, allowing it to grow into new locations. So this knowledge is going to be really useful. I want to turn my attention now to another story in the lab, those looking at onion bulbing. And again, it's similar. People and John McCullum at Plant and Food Research and onion growers were really interested in understanding how a plant becomes adapted to new areas and how you can breed an onion suited to the right conditions. And there was a particular problem with premature flowering. Some wise man said once that to be a great onion, you need to be adapted to your environment. And so onions grown in more tropical areas where the day length differences are shorter will flower with just 10, 12 hours of day length will trigger bulb formation. So an onion bulb formation is caused by changes in day length. Whereas if you grew an onion in Dunedin, then it will form a bulb after 14 hours of day length. So you need different varieties for different regions matched to their location. So there's different types of breeds needed. So what underlies the difference between those breeds? And you certainly can't grow a short day onion in Dunedin. It will form a bulb way too small bulb and flower prematurely, and it'll be a disaster. So what is the difference? So we set about to try and understand the process of flowering in onion and inadvertently probably bulb formation in onion. And here I'd like to acknowledge the work of Robin Lee in my lab. Robin's worked for me for 10 years, slightly over 10 years now, and basically runs the lab, and I really couldn't do without her, and her help with the supervising of the students and helping me is absolutely crucial. So to be able to keep her in the lab, the pressure of getting money has luckily come through, but certainly without these key individuals, you would struggle. Because I was working on onions, Jiffin, who had a family farm in India growing onions, came and wanted to work with me, do a PhD, so he has been a tremendous asset. He's now gone back to a university position in India. And Mo, who also worked on the project, is now a breeder in Pukekohe, an onion breeder. So I'm very proud that the skills that they've been learning are now have some more practical use. And John McCullum, there's not a man on earth that's more passionate about onions. So using some of this high throughput sequencing technology, in fact, in Joe Stanton's machine and anatomy, right in the early days, we were able to sequence the messenger RNA from onions and for the first time try and work out what genes were doing what. And what we discovered was that there was an FT gene that triggered flowering, so that wasn't surprising. But what we were surprised at, there was an FT gene that triggered bulb formation. So in onion, then FT is the hormone that triggers bulb formation. And when we made a genetically modified onion that could do it in Lincoln for us, we got, to our surprise, onion bulbs forming straight away right in tissue culture. So that was a very exciting day when we saw those, proving that FT gene really does turn on bulb formation. So what's happened in evolution is there's been kind of a split of the pathway, duplication of these two FT genes, one keeping the day length perception role, but then turning on to do something different, turn on and make bulbs, whereas others, one is just turned on in the cold and triggers flowering. So we've been working away trying to understand the other steps in the pathway and what the genes encode and how these all work. And the key thing we're trying to discover is really what is the difference between an onion adapted to one region and a different region. And so, amazingly, in tropical regions, the FT2 gene is turned on with about 20 degrees as a vernalization period for an onion, a cold period, whereas we need sort of 4 degrees for a New Zealand onion. So how the plants respond to different temperatures of cold, how they respond to different changes in day length, and how that's occurred is what we're wanting to work out now. And hopefully, at some stage, we'll be able to tell the breeders what genes to breed in to get them adapted to different locations. Okay, so another story I wanted, I'm probably running out of time a little bit, never mind, is vitamin C production. And so, again, it was through a collaboration with plant and food research, and I've got a couple of students, Matthew and Erin, who did some of the work that I'll talk about. The thing about vitamin C is a little nutritional fact for you, that vitamin C helps you absorb iron. And iron deficiency is a major problem around the world. So I think if we can breed crops that have higher vitamin C production, vitamin C levels, it will be a tool to help people uptake more iron from their diet. And probably need to do that as we eat less meat and more vegetables and fruit. Okay, so through the work of plant and food, then they found that they'd worked out the pathway for vitamin C production, and they knew this enzyme here, just call it GGP, is key rate-limiting step in the pathway. So here I've got the gene making this enzyme, which makes vitamin C. And a bit like the example I had with FCA, that flowering time gene that was regulated in a feedback loop, what we've found was that in the case of this gene, is that there is a sequence upstream that actually encodes a little peptide that when there's high levels of vitamin C, we think, some evidence that vitamin C actually binds to this peptide, stops the translation, stops therefore this downstream protein being made, and therefore reducing vitamin C. So an elegant feedback mechanism to make sure you have the right levels of vitamin C in the plant. Some plants, though, have huge levels of vitamin C, tremendous levels of vitamin C in these superfruits, 100 times that of an orange, for example. And there's this non-commercial kiwi fruit variety, again, has 20 times the level. So we're really interested in understanding how that's happened. And again, RNA sequencing is one tool that we've used to try and figure out what's going on. And it turns out that they have extremely high levels of these messenger RNA encoding these key enzymes in the pathway. And so what we think is probably there is some factor, transcription factor, that's turned on at high levels that can turn on all these genes. And we've also shown that there are changes, not complete mutations, but changes in the way this feedback regulation works to allow more of the enzyme being made even when there's reasonably high levels of vitamin C. And using that knowledge and gene editing, we've been able to create Arabidopsis plants that have 3 times more vitamin C by mutating that regulatory sequence. And we've got genetically modified tomatoes that have 5 times more vitamin C by doing both, turning on the gene, actually just in the fruit, and not having this feedback regulation. So that's the highest vitamin C level that's been found in a tomato. But still not near where the limits are. So I think there's other feedback regulation going on that we need to discover to get it even higher, but this certainly would make a big difference if we could market that tomato. Okay, finally, I want to talk about the work that we're doing with ryegrass. So this is the father-in-law's farm with beautiful ryegrass growing just over the Paiotua track from Palmerston North. On my side of the family, there is some farming in there. I had to delve a wee way to find some farming in my side of the family. And I'll talk about my great-grandfather, who was born in 1819 in Scotland. And he studied at Edinburgh University, and then, aged 22, set sail for Australia, arrived in Port Phillips, and bought a farm, and ended up specialising in breeding short-horned cattle and merino sheep there, and became quite an expert breeder, and was intrigued about the value of inbreeding. He thought that there was huge value in inbreeding and the way that they bred, and even wrote books on breeding, one published on the true principles of breeding in 1865. There was another Charles who was also interested in breeding, a wee bit more famous, I know. He was born 10 years earlier than my great-grandfather, but Charles Darwin also studied at Edinburgh University, also went on a voyage, again a little bit more famous, and again wrote a book on breeding, the variation in animals and plants under domestication. And in that book, very pleased to see that he referenced a couple of my great-grandfather's works in this book. So what Charles Darwin had noticed and says in that book is he noticed there were 2 types of plants. Some plants which naturally self-pollinate so that they inbreed, and some plants that naturally outcross and can't inbreed. And a couple of years after writing that book, he also noted that plants that outcross have greater height, weight, fertility than their self-fertile counterparts. So there's actually advantages of both breeding strategies. The advantage of a pure breeding line is that it's purebred, and whatever you plant is going to be uniform, whereas an outcrossing plant, you end up with a mixture of phenotypes, a mixture of plants. But there is this potential to get this hybrid vigor, as it's called. And combining the advantage of those 2 breeding systems has really been one of the reasons why we've fed the world. The ability to, in maize and rice and many other crops, to inbreed plants, so you get nice uniform plant, and then outcross them so that you take advantage of this hybrid vigor, but then all the seeds, because these 2 lines are inbred, are genetically uniform, allows this high quality F1 hybrid seed that you can plant. And here we show the results. In yellow is when they started to use these hybrid varieties, and it provides a way of really accelerating the selection and breeding, and led to this tremendous gain in yield. Unfortunately, in ryegrass, yield gain has been a real struggle because it's an outcrosser, and you end up with a population of plants. And actually some of the leading varieties, just talking to a breeder on Friday about it, and he was saying that in one of the leading varieties that they still sell, he bred in 2000. So 20-year-old varieties are still the leaders. So they really aren't making the gains that we'd like them to. And so have a project, in collaboration with Lynette Brownfield, who's I share a lab with in the Biochemistry Department, Rowan, a talented postdoc who actually did a PhD with me first, went to Cold Spring Harbour and has got excellent training. Others in the lab, Shiny, Caitlin doing a PhD student, Samarth, who's another postdoc in the lab. And we're together trying to use our knowledge that we've gained from, and the world has gained from studying these other cereals, rice, etc., and how they work, and applying it to ryegrass to enhance the breeding. And so what we're working on is an F1 hybrid breeding system where you can get self-fertile plants, convert ryegrass to be self-fertile so you can inbreed, and then switch them to outcross so you can take advantage of this hybrid vigor. We have another project that's involved in trying to prevent flowering in the field because that increases production, and just induce flowering for seed production. So there's huge potential. I end really, this is my last slide, end saying there's huge potential in using our knowledge that we've gained from understanding how genes work and these systems to apply them to crop plants to enhance production. And back to my point that we really are going to need to make these advances if we're going to continue to have a productive agricultural sector without having increased land and pollution. And on this project, I'm pleased to be working with seed companies, AgResearch, Otago Innovation, MBIE, all funding some of this work, which is really exciting. And finally, too many to mention, but I'd like to thank all the students that worked with me and for me over the years, and the other people that worked in the lab. And I'm sorry that I've used more time talking about it than I should have, talking about all the work that's going on, but I haven't only touched the surface of and haven't been able to talk about many contributions of many people. And I've worked with people throughout New Zealand and elsewhere in this work, and as Frank said, I was very lucky to be funded for many years by the Marsden Fund. So thank you very much.
Kia ora everyone, I'm Sally McCormick, the HOD of Biochem, and it's my job, I've got the easier job of thanking Richard tonight. So ngā mihi Richard, thank you for giving us that wonderful insight into your research endeavours over the last 20 or more years. Those of you that are avid gardeners will know the importance of the right timing of flowering. But of course, Richard has dissected that right down to the molecular level, understanding the genes and the pathways involved, and now having the ability to use that knowledge to be able to manipulate flowering in a whole range of plants. And so that has huge potential, along with the vitamin C work he's mentioned, to enhance nutritional value of plants. You can see how that might have global implications. So great stuff. I just wanted to touch on a couple of things with Richard I think has been, it's quite unique, two qualities I think quite unique, and they've come through both in his talk and with Frank and talking about them. One is ability to engage in new technologies, and I think that's been true all the way through. In fact, I was reading one of the referees' reports for his job application in 2001, and it stated that he was the best cloner that had ever been in the lab. I suspect that was from Carolyn. Now, cloning wasn't new back in the late 1990s, but it was certainly very, very difficult. And these days we have kits that make it a lot easier. So clearly, technically apt and has maintained that engagement in new technologies as they've come aboard. So there's been a lot of new gene technologies come aboard in the last 10, 20 years. And he's engaged in just about every one of them. The next generation sequencing, microarrays before that, the gene editing that is available now. And he's made sure that all of his lab people are very well trained in those technologies. The other important quality I think he's shown, and again, right from the very start, his foresight to engage with the end users of his research. So the CRIs, Plant and Food, the plant breeders, right from the very start, he has had engaged with them and has had some very fruitful and long-standing collaborations with them to this very day. And I think those two things have, and many other things have come together to ensure that he has had very long-standing stream of funding, and Frank alluded to this, both from the basic end of funding right through to the applied contracts with plant breeders and things. So we are very proud of you, Richard, and we are very happy that biochemistry is your home. You have had a long stream of students coming into your lab, more than most, and again, I think that also plays to your role of being a very passionate and engaging teacher. Students love you, and they want to be working with you. So with no further ado, big congrats, and I think under here we have a wee gift for you. It's a hidden gift. So there we go. So I think it's back to Pat.
So I'm now the acting Pro-Vice-Chancellor for Health Sciences, and to finish off this evening, I'd like to echo Sally's thanks to Richard Macknight. That was a really tremendous talk. I thoroughly enjoyed it. I shall check my garden more thoroughly in future. And I'd like to thank everybody in the audience for coming to this wonderful talk. And I'd now like to invite you all over to the Staff Club for refreshments and the ability to talk to each other and to talk to Richard and to talk to his wide family. So thank you for coming.